So welcome to the last, but of course not least session of the SMI 2021. And the session was organized by Rajarshi Guhaniyogi. Unfortunately, he couldn't attend the session today, so he won't be participating, but I'm chairing. My name is Jaroslaw Harezlak. I'm a professor at Indiana University. And we're very happy to have three excellent speakers in the session. So without further ado, our first speaker is John Kornak, and he will talk to us about Bayesian image analysis in transformed spaces and the computational packages implementing the methods. John, the floor is yours.
Thanks, Jarek, and I'm glad you were the one that had to say Raj's name, not me, because that's pretty tough. But yeah, it's unfortunate Raj can't join. I'm really grateful to have that he invited me to the session, and I'm grateful again to all of the organizers for doing a really awesome job of setting up this meeting online, and hopefully next time in person. So I'm going to talk about Bayesian image analysis in transform spaces, mostly focusing on Fourier space, but a little bit on wavelet space. There's quite a few people that have gotten involved with the project over the years. Carl Jung, Russ Boylan, and Eric Friedman are the three most notable that have played a big role. So I'll start by talking about conventional Bayesian image analysis. I'll go through that for the first half of the talk. I'm going to go through pretty quickly because I've covered these before, but I need to cover them to get to the new parts at the end. Bayesian image analysis in Fourier space after that, so that's reformulating the whole thing into Fourier space. I'll talk about the Python package we have to use BIFS, and then talk about, I guess my link to the supervised part of this session is that I'll talk about a data-driven approach to applying BIFS and an example there, and then I'm going to move into wavelet space, and I'm going to skip lots of details, but if you can ask me all about them at the end. So conventional Bayesian image analysis, you have a typical setup where you get to observe the image Y on the right here, which is the truth plus some noise. What you would really like to see is X, the truth, over on the left, but your imaging modality doesn't allow you to see that. So you have to take Y, come up with a model and try to reconstruct X as best you can. Or maybe you want to enhance X, for example, maybe you want to highlight tumors that are in the image, and so you'll focus on trying to get an image that highlights the tumors. So the usual Bayesian image analysis formulation, you have a likelihood, which is your process that takes your model for the process that takes you from the truth to what you actually get to observe. So that's your noise process or your degradation process. You have a prior for the characteristics of the image a priori. And if you're not familiar with Bayesian image analysis, we'll you'll start to see what these prior characteristics are about, but it's features of the image or aspects of the image of the truth that you want it to have. So in particular, you want it to not have noise would be a first step. And then using Bayes' theorem, we get a posterior estimate from there. For example, if you try to go to the maximum of the posterior distribution, then you get the maximum a posteriori estimate or the map estimate. To date, most work or pretty much all work has been in image space in terms of defining these priors. So you have, as you know, if you work in images, your pixel or your voxel is your base unit of data. And these models kind of talk about the relationships between the pixels in space. And for a lot of these sort of common models, particularly the Markov random field model, you talk about neighborhood structures and how one pixel influences its neighbor and vice versa, and you get this interdependent structure. And in a very loose way, you can think about this as being some kind of probabilistic smoothing. So, and if you work in fMRI, you would know that you're always advised to smooth the images with a kernel before you start. So In this case, instead of just blindly smoothing something, you're kind of trading off between the data, what you got in the data, and these prior characteristics of smoothness that you might want, for example. Okay, as I said, we're going to work over in different orthogonal spaces, and I'm going to start with Fourier space, and that's what we started working in originally, and it really, you can do a real lot with this. Here's just a cartoon of Fourier space, or at least 4 locations in Fourier space that you see marked on kx, ky coordinates. And you see that each of those locations, I've got a little pictorial image. So at 0, 0, it's flat. At 0, 4, you've got 4 horizontal bands. And these are two-dimensional sinusoidal functions. And basically, when you represent something in Fourier space, you are representing it as a linear combination of these basis functions, of these little images. You put them together as a linear combination, and you get the image that you started with. And the key concept here is that each of these little basis functions is a spatially correlated process in its own right. So instead of having to model spatial correlation between neighbors in an image, I'm going to try and do it through these basis functions and treat them independently. So the way the formulation goes is you take your image data, you transform it into Fourier space, and then you apply Bayes' theorem over there. So this looks the same as the equation that you have when you're working in image space. The posterior for the Fourier transform of the truth, given the Fourier transform of the data, is proportional to the prior times the likelihood, all working in Fourier space. That's what this little script F is telling, is that it's the Fourier transform of whatever is after it. And by modeling this Fourier space independence, by saying I'm going to treat each of the Fourier space locations independently in my model, if I look at the Fourier transform of X at a particular location in Fourier space, given the complete Fourier transform of the data, it only depends on the Fourier transform at that location K. So if I want to maximize this whole thing, now the posterior distribution of the Fourier transform of X given the Fourier transform of Y, or I want to sample from it, I don't have to deal with it as some large, complex, interdependent structure, as a big, complex, multivariate posterior distribution. I just have a whole bunch of little independent problems. And if I solve the independent problems, I put it all back together, inverse Fourier transform, and I'm done. Okay. So that's the idea. And in terms of why we're taking this approach, and here's sort of like I want to motivate how we build these priors in Fourier space. At the top here, I've got an image, and all I'm going to do here is decompose it into different sets of spatial frequencies. So over on the left, I just have, you might be able to see just the central tiny point here. That's the mean image at 0, 0 location. Here I have a bunch of low frequencies around the origin, and you see you get like a sort of very smooth representation part of this image. I go to higher frequencies and I start to get sort of the middle size objects jump out. And then if I go to the really high spatial frequencies, I get the little details that you see in this right-hand image. So the idea is now, how do I define my distribution over all of these spatial frequencies in a way that represents what I want my prior distribution to do? And again, just like in the Markov random field, I can think of this as being sort of like a probabilistic smoothing thing. But in this case, instead of thinking about kernel smoothing, I'm thinking about frequency bandpass filtering instead in some way. And I'm going to build a prior distribution over all different ways of, I guess, smoothing. So to build the prior, 2 steps you need. The first step is to specify the prior distribution at each location in Fourier space. And then I can't do that for each and every location. So I need to specify some way of how these parameters are going to vary. So a picture will help here a little bit. So here's my representation of Fourier space laid out flat, kx, ky. And at each point, I need to place a distribution for my signal. So here I have something that looks a little bit like a log normal distribution. So it has a location parameter mu and a scale parameter sigma. And then what I need to do is define a surface over the whole of Fourier space, or how I'm going to specify my mean parameter. And then I do the same again for how I'm going to specify the standard deviation parameter. Okay. And in case all of that got lost, here's a broad schematic of the whole thing. We have an image with noise that we start with. We want to work in Fourier space, so we Fourier transform it into Fourier space. We have a noise process with model in image space, but we want to think about that in Fourier space. So we're over with the FFT there. And I have a prior that I'm just saying, this image just saying I prefer smooth images to non-smooth images. And in Fourier space, that corresponds to putting a lot of mass in the center of Fourier space. All of that gets fed into Bayes' theorem. From there, we extract an estimate that would sit at the maximum of the posterior distribution or the mean of it or something. And then I just inverse Fourier transform and get my reconstruction. So we have a Python package out there to do this, and it was all nice and working. Unfortunately, something's just broke, but it should be fixed very soon. And if you want to play with it, I'm really open to discussing any problems you might have. But basically, you just go to GitHub, you grab it. There are two main functions. There's a GUI function that you run. If you have Anaconda installed, which is kind of like a big Python wrapper with all of the math and numerical libraries built in, then it runs pretty smoothly. And I'm hoping that I can convince John Micheli to put an R wrapper around the whole thing and put it into Neuro Conductor. Or maybe I'll have to do it if not. But basically, if you work with the GUI, it looks a little bit like this. You get this panel with 4 pictures when you fire it up. You can load an initial image. And you see the image here is actually sort of that noisy MRI of a breast that I had right at the start. You then go into these parameter sets, set various parameters, like for the prior distribution, I want it to be Gaussian with a certain scale. Then for my k-space parameter function, that's how the parameters are going to vary over Fourier space. I have that decaying away from the center of Fourier space with an inverse power law, and the inverse power here is 0.5. I have to define my likelihood distribution. I'm just going to define that to be Gaussian too. And then you just go up here, you hit get map estimate image, and you get a result out. And you can see here the initial image, reconstructed image. You get the Fourier space versions of it. And over here was the original truth before adding in the noise. And you see, we sort of, I haven't sort of played with this very much to try and get any better images. But it's recovered some of this stuff here a little bit, whereas it's completely lost in the initial image. Perhaps it's over smoothed here in this region, so you might want to sort of look for a prior that doesn't smooth as much there. But overall, does sort of reasonable job. Here's another just image. This one I did play around with a lot to try and get a nice reconstruction. You can see that really do a pretty nice job of pulling out an image out of something that looks close to complete noise. Okay, so that's the standard application of Bayesian image analysis in Fourier space. I've talked about that quite a few times before, and now I'm going to sort of slow down a little bit and talk about two different things. The first would be like the data-driven supervised version of Bayesian image analysis in Fourier space. And then the final thing I want to talk about is moving over to work in the wavelet space. So here, when you're working with this supervised form, you don't define that parameter function anymore about how the parameters of the prior are going to vary over Fourier space. Instead, I want to use a database of high quality images to kind of supply the information for how to build that prior. And then over that database, estimate what the distribution is at each Fourier space point, making again the assumption that over Fourier space, I can assume independence because the spatial correlation is built in. So you can estimate the distribution at each Fourier space point in two ways. You can either completely use the empirical distribution, or you can kind of build parameter functions just by taking the estimate of the mean and the standard deviation, for example, at each and every location in Fourier space. So you line up all your images across all subjects, and then you build the prior at each point in Fourier space. And in the grant that we had this example, we had the project that we wanted to use perfusion MRI kind of as a surrogate for PET. And I didn't realize what we were getting into when we picked this project, but I kind of assumed that they were trying to measure the same thing, but they're really not. PET measures glucose level in the brain, and perfusion MRI measures blood perfusion, blood flow in the brain. And PET is routinely used for dementia diagnosis, but it's a bit of a pain. You're going to have to get routine MRI images anyway, so to have to send them separately to a PET scanner to get the PET image is A, expensive, B, you have to inject them with some kind of nuclear tracer. Just overall, if we could avoid the PET, it would be great. But it's really just a loose association, a loose correlation between the blood flow and glucose levels. It's highly correlated, but that depends on your definition of highly correlated. And the objective is what we would really like to do is be able to imitate the PET using perfusion MRI. And that way we don't need to send the subject off to the scanner. And you can see how far away we are here really before we start. So here's your original PET scan, and here's an original ASL image. And clinicians are used to reading the image on the left. They are definitely not used to reading the image on the right. They're not looking for very much in here, by the way. They're just looking for kind of imbalances from left to right. You can see over here it's darker on this side, and this is kind of indicative of frontal temporal dementia. Okay, so our idea was we'll take a whole bunch of PET images, we'll build a prior from that, and then use that on ASL images to try and get them to look more like PET images. And hopefully they'll capture the part of the PET images that the clinicians like without removing any information about the diagnosis, which is what we want. Here's an example of where we end up. Now, here's the original PET, here's the original ASL, and over on the right is our data-driven BIFS formulation. So we do see some of the gradation from left to right that we lost here. Now you can see these are supposed to be co-registered images, but you can see that the co-registration is not great between this PET and ASL. The clinicians at first sight like it, but we need to do now a sort of full validation exercise where clinicians are sort of blinded to diagnosis and see if they do as well with these perfusion as they do with ASL. Yesterday, yesterday or the day before, Dana gave a talk where she got these clinicians to validate her work, and I was like super impressed because it's really painful trying to get these people to find time to look through a stack of images for you and grade them. Okay, so a little summary of some of the benefits of BIFS. it's fast because we have this independence property over Fourier space. It's really fast and it's easy to parallelize. It makes it easy to be able to, the whole approach is flexible, so it's easy to try different priors. Unlike in conventional Bayesian image analysis where you have to write a new set of code, here you just slot in a different parameter function, you have a different prior, and you can try things out. We get isotropy for free if you want, so that the correlation in each direction is uniform. That's hard to get in a Markov random field because you have preference for up and down directions. And we have a kind of resolution invariance. If you change the resolution of an image, if you have images with different resolution, you can't use the same Markov random field model. You have to tune the parameters to approximately match the spatial correlation. When you're working in Fourier space, you just slot your lower resolution frequencies into the middle of the higher resolution frequencies, and you're good to go. This is so fast that we could be using this as something like as a smart spatial smoothing in fMRI instead of hitting things with a kernel just to be able to apply Gaussian random fields and stuff. And instead of losing features that we might be interested in. So I would encourage you to sort of try that out. Jarek, do I have any time left or? 2, perfect, 2 minutes. I can do this in 2 minutes. So wavelets is what we've been working on most recently. So Fourier space is one orthogonal set of basis functions. Now we want to look at other ones. Wavelets have a lot of potential benefits. There's a lot of different representations. You can see it here over the right. There's different kinds of wavelets that you can have with various levels of complexity. The rule that they have is that they have to sort of like integrate to 0 and they have to have compact support. And then you take these and you build your basis set by shifting them in space and scaling them. And the different wavelets have different properties. Some will be better at detecting edges. Some will be better at capturing other kinds of structures. One little advantage with these over Fourier space is that if you have Gaussian noise in your image space, it translates to Gaussian noise in wavelet space. You can model local objects with wavelets that you can't do with Fourier space. With Fourier space, you're working across the whole frequencies averaged across the whole image. And finally, you can get at textural properties. I'm not going to go into these exponents, and I don't think I could do a good job of it anyway. It was like my colleague Carl Jung who looked at these. But you'll see in the example result here that it can be quite powerful. So over on the left here is an original image. It's made up of 2D fractal Brownian motion plus these semi-transparent sort of figures on top of that object. And then you can see he added in quite a lot of noise over here so that you can hardly see anything, and then applied the conventional thresholding wavelet methods to look at these that try to get at the textural properties. And then this is our Bayesian method looking at across wavelet space. And it does okay at getting some of these objects back. And in particular, it does a good job at capturing the sort of textural structure here. So it's modeling the textural structure across different scales. And by doing so, we're able to, it's able to pull back this little bit of a sort of a cloud here to some degree. So we're sort of like really impressed with how that's going. And we don't have the package out for this yet, but we hopefully will soon. So in conclusion, working in Bayesian image analysis transform spaces is a really powerful option for Bayesian image analysis. Super fast, has nice properties. You could even use it for smoothing long time series because it's so fast. the software is available on GitHub. The command line package is more powerful, but if you just want to play with the GUI, please do so. And you can email me when you come across any inevitable problems that might occur. And I should acknowledge the grant that I got for this. So thanks very much for listening. And I'll take, I guess, questions at the end and let Jarek pass it on to the next speaker. Thanks.
Okay, so thank you, John, for the great talk. And I also want to thank the organizers for organizing this great conference. And I'm also thankful to get an opportunity to present my work. So today I'm going to talk about Bayesian regression with undirected network predictors with application to brain connectome data. This is joint work with Professor Abel Rodriguez at the University of Washington. So at the very beginning, I would like to talk briefly about networks. So a network consists of interrelationships among a group of actors or nodes, as they are called. And on the left-hand side, you can see an example network. And a network could be directed, in which the relationship between any pair of nodes would be asymmetric, or undirected, in which this relationship is symmetric. Also, a network could be binary, in which we just care about whether there exists a relationship or not, so 0 or 1. Or it could be weighted, in which the relations come in the form of weights or counts, for example. And the networks that I'll be working with would be undirected and weighted, which is why I've marked them in bold. And on the right-hand side, you can see what is called an adjacency matrix, which is a really popular way to visualize a network. And so here, again, there's an example network that I'm showing you that has 5 nodes. And it's an undirected network, which is why the matrix is symmetric. So the lower triangular region is going to be the mirror image of the upper triangular region. And we do not model self relationships, which is why the diagonals are set to 0. And also, any cell in this matrix is what is called an edge that connects the corresponding nodes. So, for example, cell F is the edge that connects nodes 2 and 4. Okay. So with this little introduction, let me move on to the motivation of our work. Now, we have data on multiple individuals, and for each individual, we have 2 pieces of information. First, we have their brain network, which has been collected using diffusion tensor imaging, which basically measures the number of neuron connections between different regions in the brain using restricted diffusion of water in the brain tissues. And so each individual has a brain image, and the brain can be viewed as a network. So in our case, the brain has been mapped according to the Desikan Atlas, which divides the brain into 68 different regions of interest. And so the brain can be really visualized as a network having 68 nodes. So it can be visualized using this adjacency matrix for every individual of dimension 68 times 68. Okay, so that's the dimension of each brain network. And, it's a symmetric network because the symmetric matrix because the network is undirected. And also, these little colors that you see here, they are basically the number of neuron connections in that edge corresponding to, the pair of nodes, okay, connecting that edge. So these networks serve as our predictors, and the response that we have is continuous in nature. It is called the Composite Creativity Index. It's a measure of creativity of an individual, and this or CCI for short. And this Composite Creativity Index was proposed by Rex Jung et al. In their 2010 paper on creativity. And the way it has been collected is the following. So the subjects in the study, their creative works have been collected and graded independently by 3 judges. And, those grades have been assimilated together to form this composite index, that is intended to measure the creativity of the individual. Okay, so we have this network predictor and a scalar response. And so given this, our important goals in this study are the following. So we would like to predict the scalar response, which is composite creativity index, based on a new undirected network predictor. Okay. Secondly, we would like to identify active network nodes or regions of interest in the brain impacting the response. And third, we would like to identify important edges or interconnections between these regions. Okay, so these are our three important goals in the study. Okay, so given this setup, one thing that we can do right away is, we can take the brain network, which is the adjacency matrix, and we can break it up into a long vector, and we can stack these vectors together to form our design matrix X, and we can simply regress our outcome on X. Now, I want to mention that we are in the realm of high dimensional regression here. Because, for example, if you think of our real data, the brain network, the adjacency matrix is of dimension 68 times 68. Okay, so if you count the number of elements in the upper triangular region of such a matrix, it's going to be around 2,500. And the number of samples that we have is just about 100. So the number of predictors is much higher than the number of samples. So, if we took a frequentist approach, we could use the lasso, for example, or some of its variants. Or if we use a Bayesian approach, we can use some kind of shrinkage prior, like the spike and slap prior, or some kind of, continuous shrinkage prior, like the Bayesian lasso or the horseshoe, for example. Now, taking this approach has its limitations. So we are breaking the network down into a long vector. So we are really destroying the network structure, and hence we would really be unable to identify the important nodes. And, I would like to reiterate that node selection is an important goal for us. And also, our interpretability remains limited to individual edge selection. Okay. So given this background, let me now introduce to you our model, which we call the Bayesian network regression model. And it is a high dimensional regression model of the continuous response YI for the ith individual on the undirected network predictor AI. Of dimension V times V. Now, this AI that you see here, it is, the adjacency matrix for the ith individual. And V, as I mentioned, was 68 in our real data. Okay, that's the number of nodes that we have in the network. So this is our model, number one. And, B here is the network coefficient matrix of the same dimension as AI, that's V times V, and its kth element is given by beta KL. And, this brackets with the F underneath is the Frobenius inner product in the space of matrices, and the errors are normally distributed. And like AI, B is assumed to be symmetric and with zero diagonal entries, and so since self relationship is absent and AI is symmetric, we can actually rewrite this quantity on the left-hand side as the one on the right-hand side, okay? And so letting 2 times beta KL be gamma KL, we call gamma KL, we can actually rewrite our model as model number 2. And so, this model 2, it really connects the network regression model with the usual form of the high dimensional linear regression, with these AI KLs as the predictors and the gamma KLs as the corresponding coefficients. Okay. Usually in ordinary linear regression, the predictor coefficients are indexed by the natural numbers. But in our model, you'll see these two indices, K and L. So, so we index their positions in the matrix B. And so, using this, we can keep a tab not only on the edges, but also on the nodes connecting the edges. Okay. So that's important. And now, having described the model, let me talk about our prior structure. So we call our prior the Bayesian network shrinkage prior. And so first, let me introduce latent variable UK, okay, which is a vector of length capital R, and it corresponds to the kth node. So there are capital V nodes in all, and corresponding to each node, I have a latent variable, okay, U. And so in the context of our model, and I'll quickly go back to the model. So here it is. So you see these gamma KLs. These are the edge coefficients, and back to the current slide. So given the other parameters, the gamma KLs a priori follow a normal with this mean UK prime lambda UL and variance SKL times tau square. Now, if you look at the variance term, this SKL, these SKLs, they are called the local scale parameters, and they control the shrinkage of the coefficients. And the tau square is the global scale parameter. And, we have put an exponential prior on the SKLs, and mu log tau square is flat. And if you look at this mean term, this is very crucial, and this is where this network thing comes into the shrinkage prior, okay? So we notice that this gamma KL corresponds to the edge that connects nodes K and L, and UK corresponds to the kth node, UL is the latent variable for the lth node, and this lambda here, this is, it's a diagonal matrix. And so this term UK prime lambda UL, it really incorporates the network structure into our setup, and it in a way captures the interaction between nodes K and L. Okay, and so this prior is actually inspired by the low-rank spectral decomposition of a matrix, this form, although it is not necessarily a low-rank spectral decomposition. And we notice that this term, if it is zero, if it equals zero, then that implies that this gamma KL a priori follows the Bayesian lasso shrinkage prior, which is a continuous shrinkage prior. And the addition of this term makes all the difference to our structure, okay? It adds the network structure to our setup. All right. So the reason why these UKs have been introduced is to assist in node selection. And so we assign a spike and slap prior on these UKs. And, this is the non-zero portion, and that's the zero portion. And corresponding to each UK, we have a latent variable psi k, which takes value 1 if UK is non-zero and takes value 0 if UK is 0, okay? And, we have a Bernoulli prior on the psi case. And of course, here, delta naught is the Dirac delta function at 0, and M is the covariance matrix of order capital R times R, because, this UK is of length capital R. And, this delta here, that corresponds to the probability of the non-zero mixture component, which is this portion. And we do not fix delta, we put a prior on delta, and that is required for multiplicity correction in variable selection. Okay, and as stated, so we have a beta prior on the delta, and theta square, which was a parameter in the exponential distribution, that has a gamma prior, and M has an inverse Fisher prior. Okay, and if you recall that term UK prime lambda UL in the mean, that lambda, as I had stated, that is a diagonal matrix. And the diagonal elements can take value 0, 1, or -1. So basically the absolute value can be either 0 or 1. And the sum of the absolute values of this diagonal matrix is called the effective dimensionality of the latent space. And in order to learn the effective dimensionality, we assign a multinomial Dirichlet prior on each of the diagonal elements. And we note that the posterior probability of the event that UK equals 0 is going to be large if the kth node is inactive. And, in a way, the posterior probability of UK equal to 0 also gives us the uncertainty related to node selection, okay? All right, so that completes our prior structure, and I will quickly now go on to some results that we have got using simulation studies. And in our paper we have a lot more results, lot more settings. So if you feel interested, please feel free to take a look at our paper. But in the interest of time here, I will go through only limited number of cases. So I show you here 5 simulation cases, okay? So cases 1 to 5. Here I'm varying the node sparsity parameter. So node sparsity is basically, in the truth, what is the probability that a node is inactive? As the data has been generated. So basically, if the node sparsity is less, it means that the underlying network structure is stronger. And if the node sparsity is higher, then the underlying network structure is weaker, okay? And here we have some competitors that we are comparing our model to. So this is our model, Bayesian network regression. And here is the lasso, the ordinary lasso, and the Bayesian lasso, and the horseshoe, and another recent paper, which it is a frequentist approach to network regression also. And here we can see that our method, you can see in all these settings in terms of the mean squared error of the edge coefficients, which are the gamma KLs. You can see that the mean squared error of our method is, we are outdoing its competitors by quite a bit. Although I would like to point your attention to, case 5, where you can see that the mean squared error of our method is actually, it's quite close to the competitors, okay? And the reason here is that if you look at the node sparsity level, the node sparsity here is much higher. So it means that the underlying network structure is weaker. And so, our method's edge over its competitors, it diminishes, okay? All right, so now I'm going to show you some results from node selection, for node selection. And we have, again, 5 simulation cases that I'm showing you. And we have 20 nodes here in our simulation setting, V equal to 20. And if a box is blue, it means a truly active node, and a white cell is a truly inactive node. And the posterior probability of a node being active has been overlaid. So you can see that in almost all the cases, so if you look at the truly active nodes, you see that the posterior probability is actually quite high, corresponding to the truly active cases. So and it's definitely greater than 0.5. So we are, if we use the median probability rule, that is, if the probability is higher than 0.5, then we classify a node as being active, then we are doing it correctly for the truly active nodes, okay? And for the truly inactive nodes, you can see that the posterior probability is quite low. So, we are doing quite well in terms of node selection, although, that also is affected somewhat in case 5, where, the node sparsity is higher, although the classification remains the same, but the numbers are less extreme, okay? Okay, I'm sorry, so I'll just close the chat. All right, so that was simulation study. So now I'm going to talk about the data motivation. And, I already talked to you about our real data, where we have the brain networks of individuals. And the brain of an individual has been divided according to the Dataset Atlas, as I mentioned, which divides the brain into 68 regions of interest. This is one view of the Dataset Atlas, and I do apologize for the font if it's not clearly visible. But this atlas, it divides the human brain into 34 regions in the left hemisphere and 34 in the right hemisphere, okay? So 68 regions of interest in all. So we can view the brain as a network having 68 nodes, okay? And when our method has been applied to our data, actually we have found that 19 out of 68 regions of interest have been identified by our method as significantly associated with composite creativity index. Again, these names are contained in our article, so please feel free to take a look, and I do apologize for the font here. 7 regions have been found in the left hemisphere and 12 in the right hemisphere. And we have found that a large number of regions have been detected in the frontal and temporal lobes of the brain. And, some of the notable points I would like to mention here are that the findings actually, we have found it coincides a lot with some of the previous results in the literature. In particular, we have compared our findings with Chung et al. 2010 in terms of the regions related to creativity. And one thing which is important to be noted here is that the uncertainty, has been provided by our method in terms of the probability of a region of interest, that is ROI, as being active, okay? And we also do find that the ROIs in the frontal and temporal lobes, are found to have less uncertainty. So, the numbers are more extreme on both sides. That's what we found. And in terms of edge detection, we have found that 96, edges have been detected as influential out of possible 171. And, there are 10, so there are 10 frontal, and 12 temporal, and 29 frontal-temporal significant interconnections. And in terms of predictive performance, we find that our method has the lowest mean squared prediction error compared to, our competitors. It does show some better uncertainty quantification with close to nominal coverage and narrower predictive intervals. Okay. And to conclude, our method is, to our knowledge, the first Bayesian framework to propose a regression with a scalar response and network predictor. And it facilitates edge selection and selection of the active nodes, predictive of the response. And we have seen in our empirical investigations that it yields superior inferential and predictive performance over the competitors. And, we can actually extend our work to GLM settings. So a binary response, actually. So we can classify the network into 2 categories, diseased or non-diseased, high or low, etc. And that constitutes our current work. Okay. So that's all I had to say today. Thank you.
Let me introduce Dr. Shariq Mohammed, who will present his work on radiogenomic analysis incorporating tumor heterogeneity in imaging through densities. And I fully expect some music from Radiohead here when we're starting the talk. Please.
Yeah. Thank you, Jarek, for the introduction. Thank you, Raj, for inviting me to this session. It's been a great conference so far. Unfortunately, I don't have any music along with my talk, but I hope you'll still enjoy it. And thank you all for staying until the end to hear all our talks today. So I'll start with a bit of motivation about the underlying application. So we are looking at gliomas, which are a type of brain tumors, and gliomas are further categorized into subgroups. And we are specifically interested in lower grade gliomas, which are like the grade 2 and grade 3 tumors. These are the less severe tumors. There are more severe tumors as well, like glioblastomas. But in the context of lower grade gliomas, the tumor region itself is very heterogeneous. So when you look at the tumor region, the tumor tissue is sort of broadly categorized into 3 subregions: the necrotic region, which is the area of sort of the dead cells in the center. And then you have the edema, which is the outer region of the tumor, which is caused due to the tissue swelling and accumulation of the fluid. And then the region in between the necrosis and edema, which is called the enhancing core. So what we are trying to do here in the context of low-grade gliomas is that we want to leverage information from the radiology-based images and the genomic data for a set of patients to understand the interplay between these 2 types of data sets. Just to give a brief outline of what these data sets look like. So MRI images or magnetic resonance images. So we have access to these images for a set of patients from the TCGA cohort. And these MRI images are 3-dimensional images of the whole brain. So what you're seeing here on your screen is one axial slice of the brain for a given subject. And the MRI images come in different imaging contrasts. What you're seeing here again are 4 different imaging contrasts of the same brain region and the same axial slice for a given patient. So the tumor region is outlined using a red color on these images. And on the right side, you see the different tumor subregions from the tumor region on the left-hand side of the images. So as I said in my previous slide, the tumor region is broadly classified as the necrotic region, edema, and the enhancing tumor. And we have access to this type of information from our TCGA cohort. So you have these 3-dimensional images of the whole brain, and these 3-dimensional images are made up of the axial slices. We have access to the tumor segmentation labels, and then we also have access to exactly which of these pixels correspond to which of these 3 tumor subregions. So what are we trying to do here? So first thing we're trying to do is how do we examine the inter and intra-tumor heterogeneity in an efficient and biologically informed manner from these MRI images? That's our first aim. And our second aim is that once you're able to do that, once you're able to understand the heterogeneity, how can you leverage that to understand associations between your imaging data and any genomic data, so that you're able to talk about associations between molecular signatures and tumor heterogeneity. So, the outline of my talk is so on. So I'll first talk about how do we understand or how do we quantify the heterogeneity of these tumor pixels using density functions, and then I'll talk about how can we use this quantification of heterogeneity in a regression framework to understand the associations or the underlying associations between the imaging-based heterogeneity and the genomic data. So let's get into understanding tumor heterogeneity through densities. So one of the most common approaches in terms of handling imaging data when you want to understand tumor heterogeneity is to create a set of summary statistics and then use those summary statistics as features into your downstream modeling. So one of the main issues of using such an approach when you're trying to understand the heterogeneity is that there's subjectivity in the choice of what summary statistics you want to include in your model, as well as no matter like how many summary statistics you include, there's always this question about, are you capturing the entire information in this distribution of these pixel values or voxel values? So what we instead want to do is that, let's say I have this MRI images and I have the exact tumor segmentation label. So for simplicity of understanding, I'm just showing the complete tumor region here. So you have an MRI image and you have the tumor segmentation label. You can delineate the exact pixel values for this tumor, and then you can construct a simple histogram. So this histogram is giving you information about the distribution of the pixel values from the tumor region. I can smooth out this histogram and I'll get a probability density function. And this probability density function is capturing information about the distribution of the tumor pixel values. So what we are suggesting is let's use these probability density functions as our data objects directly instead of trying to summarize these pixel values. And can we using these probability density functions as data objects, can we examine variations of genomic signature through changes in these probability density functions? So probability density functions are functions. So they lie in a very complex infinite dimensional space. So we'll need tools to be able to move from that very high dimensional space to a much more tractable Euclidean representation, for example. So in our picture here, let's say we start with two different probability density functions for two different subjects. These are capturing, let's say, the distribution of these tumor pixel values from two different subjects. So these two functions belong to a very complex space, functional space. But what we are trying to do here is, can we somehow map these objects or map these data objects, our probability density functions, and use some type of a transformation so that my probability density functions can be mapped to a much more tractable space. So to do that, we use a square root transformation. And what the square root transformation for the probability density function does is that it maps my probability density functions from a much more complex space to a simpler space in the sense of a unit sphere in L2. So once you have done this mapping, what you get access to is that given two probability density functions, if I map them using a square root transformation, now I can talk about things like what's the distance between two probability density functions, what's the average probability density functions, and things like that. And that I can sort of define in the sense that if I can compute a geodesic distance between the two square root transformations, I can use that geodesic distance as the distance between the two probability density functions. So that's the basic idea. So again, like this transformation helps us it provides us tools to do a statistical task of interest. First thing is we can define distance between two probability density functions. We can compute average of a sample of probability density functions, and we can also do principal component analysis, which helps us in reducing our dimensions and moving from a infinite dimensional space to a much more lower dimensional Euclidean space. So this Riemannian geometric framework essentially is trying to map each PDF to a vector of PC scores through our principal component analysis. So the key point here is that we want to treat our probability density functions as our data objects, which quantify the tumor heterogeneity. But my framework, this Riemannian geometric framework allows us to map each probability density function to a vector representation through this PCA. And we'll use these PCAs as our tractable Euclidean representations in our downstream modeling. So what I've described now is a way to sort of quantify tumor heterogeneity from the images using a probability density function. And using our geometric framework, you can map this probability density function through a PCA to a vector of PC scores. So what we're in the context of gliomas, what we're trying to do here is we have access to 4 different imaging sequences, and we also have access to the different tumor subregions. So what we'll do is essentially look at each imaging sequence and each tumor subregion. I'll construct density functions for each subject. So, for example, in this case, I'll have if I have n subjects, I'll have n probability density functions corresponding to the necrosis region from the T1 imaging sequence. Similarly, I'll have n probability density functions from the edema region for the T1 imaging sequence, and so on. So due to 4 imaging sequences and 3 tumor subregions, I'll have 12 such groups of probability density functions. And I can look at each group separately and go through my geometric framework to map these probability density functions to a set of principal component scores. So this way I end up with 12 groups of principal component scores, which are in a sense quantifying the heterogeneity from my images. And in our case, our second aim was essentially trying to use this tumor heterogeneity or quantification of tumor heterogeneity to understand associations between molecular characteristics and the imaging data. So to do that, what we are trying to do here is we use the gene expression data available for these patients, and we will compute something called a pathway score. So to give a brief overview, a pathway is essentially a group of genes, and it contains so each pathway sort of talks about a specific biological activity, and the pathway score itself talks about how are these genes acting, are they acting in the same direction or not. But the pathway score here is something which we are interested in trying to understand associations with a specific biological activity and the imaging based features. But really you can use any any genomic marker of interest in this regression framework as long as it's continuous. So what we're trying to do is we'll use this pathway score as our response and these 12 groups of principal component scores as our predictors, and try to identify associations between these imaging features and our genomic markers. So again, the goal is to identify the density-based principal component scores that are associated with my genomic markers. We'll build a regression framework for that using the principal component scores as predictors, the groups of principal component scores as predictors, and the genomic markers are response. The two challenges here which we face is that, so obviously because we are dividing, we have 4 different imaging sequences and we have 3 different tumor subregions and 12 sets of principal component scores, you sort of need to incorporate that grouping information about the groups of principal component scores. And because you're moving from a density function space to a Euclidean space by doing a square root transformation, you also end up with a large number of predictors compared to the number of subjects. So there's also this curse of dimensionality. So again, our regression framework, you have a continuous valued response, you have certain groups of predictors, and what we are trying to do is essentially identify which of these betas are 0 versus not 0. And what that tells me is that if a specific beta is 0, that tells me that, okay, this particular principal component in some sense is not associated with the genomic score. So hence I can sort of make interpretation that, okay, maybe overall, like some characteristic of edema from T1 imaging sequence is not associated with this particular biological activity. So to do that, to identify which of these betas are 0 versus not 0, we use a group structured spike and slab prior. And this group structured spike and slab prior essentially says for each component of this beta, you place a normal prior with mean 0 and some variance. And this variance has an indicator parameter, which takes the value 1 or V0. And if a specific group, or if the principal components from, let's say, the edema region from the T1 imaging sequence is associated somehow with a specific pathway, your indicator will take the value 1. Otherwise, it will take the value V0, and V0 here is a very small number close to 0. And this is the continuous version of the spike and slab prior, and we place conjugate priors on all the other hyperparameters. So this prior structure gives us nice conditional posteriors, which come from standard distributions. So we can use GIPS sampling for to construct the posterior distribution for each of these parameters. And so what we are trying to do here is to understand which of these coefficients beta GK are 0 or not. So because of the setup of the spike and slab prior, the continuous version, you don't exactly get 0 versus non-zero map estimates in our case. So we want to, and also the map estimate will not really use the complete posterior samples to identify which of these, if a specific coefficient is 0 or not. So we use, we compute something called posterior error probability and essentially use those error probabilities by controlling false discovery rate to identify which of these coefficients is 0 or not. And I'm not getting into the details here, but I'll be happy to answer any questions about this. So to get into the, to very quickly get into the data we have. So we have 61 samples which have matched imaging and genomic data. And for the imaging data, we have 4 imaging sequences and 3 tumor subregions, as I alluded to before. This gives rise to 12 groups of imaging-based predictors. And for each of these 12 groups, we compute the principal component scores. In terms of genomic data, we have the gene-level RNA sequencing data, but as I said before, we are interested in specific pathways which are known to be sort of relevant in the context of low-grade gliomas. And for each of those pathways, we compute the pathway scores for all the 61 subjects. So given a specific pathway, you can use that, the pathway scores for that pathway across all the subjects as your response, and the imaging-based predictors in your regression framework. So here is a visualization of our results. So each row here corresponds to a separate regression, and each row is essentially giving us results for a specific pathway. So for example, the last row here is talking about the synaptic transmission pathway. And the columns here are corresponding to the different principal component scores, principal components arising from, let's say, for example, the first one here is the necrosis region from the T1 imaging sequence, and the third principal component. So this is a summary of the results across when we run our regression across different pathways. So to give a sort of summary of what we're seeing from these results is that we're able to identify several radiogenomic associations with pathways which have known to be very relevant in the context of low-grade glioma, such as synaptic transmission ion transport and other signaling pathways. For example, some of our major findings, for example, the transmission of nerve impulse pathway seems to be associated with the enhancing tumor region from the T1 and FLAIR imaging sequence. So the second row here corresponds to the transmission of nerve impulse pathway, and that seems to be associated with the T1 enhancing tumor region from the T1 imaging sequence. And that has some nice biological interpretation, which has been studied. So it's promising that we are able to identify such associations, and a deeper validation is obviously necessary to better understand and illuminate specific nuances in these associations. So in summary, what we have done here is we've constructed probability density function-based imaging phenotypes using our magnetic resonance imaging scans. And these probability density functions are essentially quantifying the heterogeneity from the tumor region from our MRI images. And we've used those density-based features in a regression framework, which we've named RADIOHEAD, using our genomic markers, in this case, the pathway scores. And we have developed a Bayesian variable selection strategies using continuous spike and slap prior to and also incorporating the grouping structure to identify which of these tumor subregions from a specific imaging sequence are associated with a given pathway, and what can you tell about these associations in the context of the biological activity of that pathway, and so on. And we have also developed a package which can be accessed through our GitHub. And some of my collaborators, Veera, Karthik, Sebastian, and Arvind, and the funding here. Thank you, and happy to take questions.